So welcome to the dialogue of the European Heart Journal today. The topic today is a very stimulating one. It's RNA therapeutics in cardiovascular disease, current status and future perspectives. And we have two outstanding guests tonight. They're both from Germany, and I will introduce them very briefly because they have a very long CV with a number of awards and publications, and so I will try to very brief. Today we have Stefanie Dimmeler from Frankfurt. She's director of the Institute of Cardiovascular Regeneration Center for Molecular Medicine at the University of Frankfurt in Germany. And we have Professor Thomas Thum, which is director of the Institute of Molecular and Translational Therapeutic Strategies at the Hannover Medical School, and also director of the Fraunhofer Institute of Toxicology and Experimental Medicine. So hello, Stefanie, hello, Thomas, and hello to Carlo Patrono, who's the co-chairman and the discussant of this dialogue, which we run on behalf of the weekly Journal Scan team of the European Heart Journal, and also on behalf of the editor-in-chief, Filippo Crea. So hi, Stefanie, how are you today?
Fine, thank you very much. Yeah. Looking forward to an exciting discussion on RNA therapeutics.
Yeah. When you were a kid, did you figure out you wanted to become a scientist, or you had other ideas?
Oh, when I was a kid, I always were very curious. So I wasn't sure whether I would become a scientist, but I certainly were looking forward to doing some things which are creative and looking for news. So I was always interested in also basic science, let's say, meaning nature and things. So it's not surprising that I ended up as a scientist. Let's say it this way.
So what happened at a certain point of your life? You jumped into science, and I'm sure you had very good mentors, and something sort of snapped in your personality, and you developed progressively outstanding science. So what happened?
I can just recall one situation. So I was studying biology, and I was having to make my decision, which at that time it was a diploma I wanted to choose as a topic. And I had the chance to either go on the ocean with a boat to look for algae, or study human platelets. And it was a difficult decision, but I ended up with human platelets. This was my beginning of cardiovascular research, I would say.
Good
so
choice.
Maybe this was the decision in the direction I was going. Don't know where I would ended up with the boat at the ocean and the algae.
Platelets, it's a good choice, Stefanie.
Maybe Carlo will ask you at some point something about platelets function, but then you developed this molecular research in your center in Frankfurt, and of course, what do you think is the status of your research right now?
I would say, at least with respect to RNA therapeutics, I think we have moved quite far towards the clinic, but still it's a way to go, I would say. So with respect to RNA therapeutics, we have started with basic science work in 2006, 2007. And now we have completed the first phase 1 clinical trial showing the safety. But of course, now it's a long way still to develop further. And then, of course, you need also success finally to really end up in bringing these therapeutics into the patients.
So Thomas, you're in Washington today at the very exciting meeting, and thank you very much for being with us. I guess this time of the day is 11 o'clock in the morning, and you're ready for the working day in Washington. So could you tell us a little bit about yourself? So how was your career developing, and how how happened that you were involved in molecular and translational science, and if you have any other comment on your story, which is always interesting to understand.
Yeah, Massimo, thank you very much also for that question. When you started to ask Stefanie about her childness or child, I already started. What could I say? So, yeah, basically, I mean, I would say I also was quite curious when I was a child, and I come from a family of physicians, basically. So I had little chance to do something different, although my mother always say, please, please, please do something different. But in the end, I also studied human medicine in the beginning, and retrospectively, I would say it was a good decision, because it's so broad. Yeah, you can develop in many different situations. So I started a doctoral thesis rather early during my medical studies, and I found it very interesting. So I always was very interested in the science, but on the other hand, also on the patient. So there was a dual track, at least in the beginning of my career, where I did both. I became a clinical trained cardiologist with 3 years in the cath lab, but also on the other hand was working a lot in research, basically, and always thought I can do everything in parallel for a long time. So, however, in the last years, it switched more and more to research. I basically stopped also treating patients now. I did that quite long in an outpatient setting. Last patients I treated was hypertrophic cardiomyopathy patients. So now I'm yeah, more or less full in the lab, and yeah, I mean, I developed my interest and love to RNA therapeutics. I think there are two stories. One is that when I started my doctoral thesis, we had one of the first Affymetrix transcriptome machines in Hanover at the Fraunhofer Institute, where I did my PhD. So I was, I mean, confronted with this possibility to look to a lot of RNA in parallel. And then on the other hand, the second one is then when I was doing my PhD in London, I read a paper about non-coding RNAs. This is something we can also speak about later a little bit. And that yeah, flashed me a little bit. It was some stories in the liver and in some other species. And I always thought, okay, that's really, really interesting. Maybe we can start to dive into that also in cardiovascular disease. And I think a little around the time also Stefanie started, we also started to dig into non-coding RNAs, and yeah, developed basic science concept, and yeah, also last year started in the meantime through a spin-off company. We founded a phase 2 clinical study in heart failure patients with RNA therapeutics. So yeah, and that is really nice, because it fits to what I really would like to do, which is translational science. So moving really concepts from the cell culture dish to patients. This is what I currently most yeah, like to do, and currently it runs well. So, but happy to discuss certain aspects further in the next half an hour or so.
So, Stefanie, yeah, your work has been always very inspiring for cardiologists, also because you cover an area which is, sort of the background of most of the things that we are doing, or we hope to do tomorrow for our patients. How would you define the approaches based on RNA in terms on how do they are they more important than other basic developments that could translate in the future in therapeutics, or you think we are already at a good point with the development of RNA-based therapies?
Massimo, I think RNA-based therapeutics, maybe I'm biased, but I would say that these are really very powerful tools and powerful strategies for the future. And I think there are several aspects. May I have some minutes to explain a bit more? One aspect is, of course, the large non-coding RNA field, meaning there are a lot of RNAs which do things in the cells which we have not understood in the past. 95% of the RNAs have not the function to generate proteins, but are non-coding, and they act in cells. And this provides really entirely novel interference methods and attempts to change chronic processes in tissues. And maybe also Thomas can allude to this later, because we were both interested in this non-coding RNA field. The second point, of course, is that with RNAs, we can, in principle, target all genes or cell mRNAs also. Meaning with drugs, you have a limitation of availability of the drug. You may, it's easier, for example, to target things outside of the cell, or the drug have to be taken up to the cell and then needs to find the target in the cell. With RNA, if you can deliver it with blocking RNAs, with siRNAs, for example, or with antisense molecules, you can block all mRNAs which form proteins. So you can, in principle, interfere with all the different building blocks of cells. And this opens a very huge opportunity. So you have a huge druggable target, number of druggable targets. And finally, of course, RNAs can be used, as we know, for vaccination strategies, so bring in proteins. So it's not the blocking, but rather the formation of new proteins. And RNAs are, of course, also the basis for gene editing approaches, because all these CRISPR-Cas strategies, which are used for gene editing, meaning gene correction, for example, or insertion of mutations or changes of the genome, also require RNA. So we can see this is a huge field which has a huge therapeutic perspective.
Yeah. Thomas, about therapeutic perspectives, what do you think at this stage are the currently available most documented intervention on RNA, and what do you think would be the perspectives in from a clinical standpoint?
Yeah, first of all, I fully agree with Stefanie that this is really a growing field in terms of RNA therapeutics from the various aspects Stefanie has nicely summarized. So we have seen the first RNA therapies quite a while ago. I think in the meantime, there are 16 or 17 FDA-approved RNA drugs. Just to, I don't want to name specific ones, but I mean those where you target specific proteins in the cholesterol metabolism are most known, of course, yeah, to reduce cholesterol, etc. That's very effective. Obviously, very effective strategy in the coding RNA field, and there are many others that are given currently to patients and are already approved. And importantly, there are many more that are currently even in phase 2 and phase 3 studies. So this number will significantly increase in the coming years. And I recently also have seen a new textbook for students. It's a well established already in students. They learn it at the medical school, and when they then become, let's say, older and be physicians, I'm sure they will be really used to RNA therapeutics, because this is such a wonderful opportunity to be very specific in terms of targeting previously untargetable pathways and genes. And currently, I can mention I'm here at the cardiovascular clinical trialist forum in Washington. This is a mixture of people from university, but also a lot of big pharma and industry is here, and also FDA and regulatory people. And nearly in every session, there is an RNA talk. So also from the pharma perspective, this is something that is really taken up. It's not that we play around as scientists in our cell culture anymore with these kind of therapies. It's really already now in patients. It's growing, and also there's a lot of pharma interest, and this will for sure, I think, boost this field. And we have seen the success, of course, with the vaccination in the COVID area. So there are many different aspects that can be developed. There are still many questions and bottlenecks. We can also address those maybe later. But I think it's still early, but there is a lot of, I think, a bright future for the RNA therapeutics.
Okay, before switching to Carlo, let me tell that we have already about 90 people and many questions already on the chat, and we will try to have our guests and the experts to answer most of the questions. But if you want to ask a question directly, you just raise your hand or write that you want to intervene in the chat, and we will unmute your microphone and you will be able to ask your question directly. So, but before doing that, this will happen in a couple of minutes. But let me ask Carlo Patrono if he has a first general question that he wants to address to our speakers.
Well, as a clinical pharmacologist, I'm always interested on one side about mechanisms, but also concerned about long-term safety of new therapeutic approaches. So we do have some safety record in the case of RNA vaccination against the SARS-CoV-2 virus. What about RNA therapeutics in the cardiovascular area? And there are two aspects to this question. What is the longest randomized evidence that we have about safety versus a placebo control? What is the longest follow-up for safety after the end of a randomized trial? And second aspect is when you induce a long-lasting change, therapeutic change, let's say blood pressure reduction with this approach, are you concerned about the need for the individual patient to actually have a reversal of this long-lasting effect? And how do you address this question?
Okay, we'll start with Thomas then.
Yeah, okay. So first of all, the longest experiences with RNA therapeutics. I mean, if we think about, again, the trials that have been done in the field of siRNAs for reducing cholesterol, for example, they are 6-7 years old now. So obviously there's a long-term follow-up already in those patients. There were not a lot of safety concerns. However, over the last years, there was also a lot of progress, I would say, in the kind of chemistry that was used for RNA therapeutics. So there was a phase 10 years ago where chemistries were used, and I'm not an expert there, but I know that certain chemistries resulted in side effects, specifically in the liver, with an increase in liver enzymes, etc. So the newer chemistries we are using are much better, but of course it's very important because it's still a new phase for RNA therapeutics, and I think we have to look for chemistry related side effects and also for target specific side effects. I think this is a difference. And of course, if you talk about long-term effects, yes, RNA therapies can have a longer half-life than, let's say, a protein-based therapy, but that also differs a little bit on the chemistry, what you're using. I mean, some of the, and you know that again from the, let's say, inclisiran field, etc., there is 1-2 injections per year with a very long-term effect, but there are other kinds of chemistry that you have to give every 2 weeks or so because the half-life is significantly lower. So it really depends on the chemistry and the target. And if you speak about blood pressure, I don't know if blood pressure is really the best target for RNA therapies, but maybe there are better ones. I don't know. But I think we are learning. There are more and more clinical trials. We are learning more about potential side effects. If you speak about in general about siRNAs and oligonucleotides, the typical side effects you have to look for are kidney and liver related, because these are two organs where you get the most uptake of oligonucleotides if you give them systemically. Inflammation is important to check. But this is why, I mean, we have to do a very proper preclinical development, toxicological studies. This is also still a new field for RNA therapeutics. And then very closely have to look, of course, in the earlier clinical developments in phase 1, phase 2 for potential side effects. And as we are learning more and more, I think we get more confidence how to use and apply these RNA therapeutics. That would be my initial comments.
Thank you. Yeah. Stefanie, any comment on this issue?
Yeah, maybe of course, I fully agree with Thomas, and I would like to add that I think the major breakthrough in reducing the side effects, which really had occurred in the early times of RNA therapeutics, is that we could now reduce the dose strongly, because we have now availabilities to target the RNAs with these either nanoparticles or also conjugations, which then reduce the necessary dose, because you directly target them to the cells in the liver where you want the RNAs to be taken up. And of course, in the liver, this is relatively easy, because the liver is relatively easy taking up substances from the body, I would say. And therefore, they could reduce the dose significantly. And this really, of course, reduces general effects of the RNA, so that the majority of side effects, at least to the best of my knowledge, are side reactions after injection, but no longer these more stronger liver and kidney reactions, which we had in the early times of these RNA therapeutics. So the dose is important. And the second, of course, is if you expect to the long-term effects, I would say, of course, it depends which target you aim at. PCSK9, for example, if you know that mutations in PCSK9 can be tolerated forever, then you are more, let's say, likely to be safe if you block this enzyme forever, yeah, or for a long time. If you have other pathogenic mechanisms or other targets, you may need to be a bit more critical with respect to the long-term effects. Of course, the long-term effects are also a positive aspect, because it's increasing this compliance. And I think this is one of the major benefits of RNA therapeutics in comparison to, I've seen the chat that why using as RNA therapeutics and not take small drugs. So of course, if you can do 2 injections and certainly cholesterol is lowered instead of forgetting your statins for 2 days and then or longer, this is of course more efficient and helps our patients who may not be always as compliant as we would wish them to be. So
before I ask Dr. Sobotka, who asked 4 questions, to take the word, because he's in the chat. But before going to him, I would like to switch this question to Stefanie from Dr. Mayo Wang. He asked whether you have any comment on potential development of anti-RNA antibodies that could affect efficacy of time. I mean, are you aware of this, or?
So I must say I have seen discussions, but I am personally not aware of antibodies against the RNA so far. And at least to my knowledge, I haven't seen data suggesting that we get tolerant against RNA. So even if it's a vaccination now, I think there's no evidence that we lose efficiency. But maybe Thomas has more insights into this. At least to my knowledge, I haven't heard that this would be a major problem.
Hey, Thomas?
I fully agree. It's debated, I would say. Evidence is more likely that it could happen when the RNA molecule is longer, right? So the smaller the RNA molecule is, that's unlikely is it that there are antibodies formed. For example, with sizes of between, let's say, 15 and 20, we have some own experiences. So there is nothing we have seen so far. So it's not very likely, but it theoretically could happen. That would be my philosophical answer.
So, Dr. Sobotka, you want to take the word? We will unmute you and you will appear on the screen. You have 4 questions, so maybe you can synthesize your comments. Are you around?
Well, perhaps what I can do is separate into 3 different domains of questions.
Very good.
The first is, could we address the purity of the current RNA preparations? My particular concern is the contaminants with double-stranded RNA related to IVT synthesis. The second is, can you speak to strategies that are underway to increase the durability of the RNA or the encoded proteins? Because as we approach cardiovascular diseases, we're talking about chronic acquired adult diseases as opposed to genetic ones. And the third is, speak to your experiences of or potential for tissue-specific targeting of the RNA, in particular myocardium and vascular tissue. So those three areas, I think, define the world of RNA therapeutics. As a cardiologist who has been inventing devices and therapies for the majority of my career and who has sat at the FDA, those three areas will largely help us define what the potential for RNA therapeutics will be.
So who's going to start? Thomas? Or?
I can start, yeah. I mean, it's a challenging but very good questions, Paul. So with the first one, I mean, I'm a bit focused on the RNA therapies we are using currently. You were asking, because they are, first of all, very important to note maybe and to say is they are very different technologies used to produce also under good manufacturing practice so that they can go into patients to produce RNA therapeutics. You were mentioning also the vaccine field, the mRNAs. Indeed, there is a possibility that impurities can occur, and there have been publications that they might be responsible for side effects, etc. I think still this is relative.
Yes? Just to, again, tighten the focus of that specific answer, the vaccines have the benefit of being one and done. I mean, so that the repeated exposure to double-stranded RNA contaminations is not an issue, but it becomes an issue when we talk about chronic cardiovascular disease and repeated exposures and maybe even larger doses. So that, to my knowledge, all of the IVT-related synthetic process for RNA are posed or faced with contaminations of double-stranded RNA. There may be other contaminants, but that's the one I'm concerned about today. So today's conversation speaks of cardiovascular diseases. We must be concerned about the purity of the preparations of RNA. And I think Carlo raised the issue in his initial.
The topic a little bit is about, but is also here antisense oligonucleotides, siRNAs, gapmers, non-coding RNA targeting LNAs, etc. And all those are not produced with in vitro transcription, but with a technology called solid phase synthesis. And here the question around purity is rather good controllable, and also there are technological advances even the last 2 years making these preparations purer and purer. So I wouldn't be concerned so much in that particular area. In the other one that you mentioned, where in vitro transcription plays a role, that might be different, but I see Stefanie also wants to add to the specific point.
Yeah, I wanted to make this exactly the same comment that, of course, if you synthesize these RNAs recombinantly, it's something else than if you synthesize those by producing them in cells also. And I would also like to say, it's not the double strand is maybe not the only problem. There are sometimes toxicities by the RNA itself, but this really depends on the sequence. And you can take a sequence which is just a few base pairs different, and then it has toxic effects, and maybe it forms some structures which are then inducing side effects, and you have to test this. So I think these are things which have to be carefully addressed in preclinical testing. And of course, you have to be safe with respect to the purity of the RNA preparation, but this is really carefully controlled. So before we went into phase 1 trials, we need to really document the purity of the preparation in detail. And there are good ways to do so. So you have to prevent, let's say, have to show that there is safety in the end. And this can be, I think, well controlled.
I don't know if we addressed all the questions of Dr. Sobolka. If we did, there's a question from Dr. Let me find it. Many questions here. Dr. Amar Reddy, how do we justify the use of RNA therapeutics when several small molecule inhibitors are available for cardiovascular diseases? Yeah, who wants to?
I think Stefanie made a good comment, right, about that already, where she said that a great advantage is the very infrequent use we have to do with these or in general RNA therapies. So it's not needed that you have a daily swallow of pills, but it might be that even patient compliance, etc., is much higher if you have a very infrequent 1 month or whatever dosing with a subcutaneous injection, for example. This I think it might be one of the major advantages. And unfortunately, so far, oral therapy using RNA therapeutics, where they can be very specific to certain targets, is not available through an oral administration so far. But Stefanie, do you want to add on that?
No, I would fully agree with you. That is a major advantage. I think also the RNA therapeutics are, the promise in the future is to make things happen which you couldn't do with conventional drugs, because as said, the therapeutic target is not druggable or is very difficult to drug with pharmaceutical strategies. So I think there is the major opportunities in the future, targeting non-coding RNAs, or also targeting pathways, gene editing for correcting gene defects. I think these are the things which I would see most promising for RNA therapeutics.
Yeah, there's a good question from Dr. Ophir Eyal. I could have made this question, but he says that the RNA therapeutic is truly exciting potential for hypertension control. However, hypertension is extremely prevalent and the therapies are very expensive, at least at this time. So this means careful patient selection would be necessary. What could guide this selection? I mean, if right now we have, some agents acting on angiotensinogen, particularly the upstream blockade of the renin-angiotensin system cascade by interacting with the RNA for angiotensinogen. And is there any clue that we can use to select people? What do you think?
So it's a very good but difficult question. I must say that most RNA therapeutics so far available are targeting enzymes in the liver. And the angiotensin system is everywhere, and you target it also. And yeah, I think this is the benefit of ACE inhibitors, that they can be broadly working. And so far, the limitation is that vascular cells itself and also cardiac cells are poorly targetable. So maybe Thomas has more ideas and unpublished insights, but the strategy we have so far to really reach the target tissue, the vasculature or other non-liver organs, is very difficult.
Yeah, maybe on the patient selection, obviously, what if you are able to develop such a compound, and then several approaches are currently undertaken in the field to produce siRNAs in the angiotensin pathways, basically, is obviously that you go for patients with uncontrollable and not anymore with standard drugs controllable hypertension, right? I mean, these would be the patients like in the hypercholesterolemia, where people started to go only to those that are completely resistant to statins or other, to specifically treat them, and the costs were mentioned. I think this is a very important point, because the costs are not so high as you would assume in this specific field of antisense RNA therapies. Basically, the production costs are rather cheap. Yeah, if you compare it to gene therapy, or let's say we have seen a lot of approaches and press releases. There was also a question in the chat about gene editing and these things. I mean, this is much more expensive to produce. The RNA, specifically the silencing RNA approaches with the current technology we have to produce is not expensive. It's rather cheap, and also the current FDA approved drugs, depending on the disease, of course. It's an orphan disease. The prices are usually very high, but the production costs are not. And if it's a more common disease like hypercholesterolemia, the price is not so high if you see that. And the production costs will go down significantly, as everywhere in the world currently, RNA production facilities are more and more invested in and are created. So there will be, I'm quite convinced that the production costs will even go significantly down from what it is now. Yeah.
As a difference with the treatment of lipid disorders, the treatment of hypertension has something to do with the blood pressure, which is an essential component of our physiology. So as Carlo Patrono said before, we may need at some point to reverse the effect of this treatment if it lasts for a long time. So for instance, coming back to hypertension, what is the policy today to ensure people from this point of view, in the case they need to have a higher blood pressure than what is achieved with this treatment?
Yeah, I think this is a difficult question, and I think, and this is why I mentioned that in the beginning, I personally think blood pressure is a not so easy thing to target. I think we have to wait until also more clinical evidence in the clinical trials is there, what is really the range and how many, let's say, side effects in terms of hypotension, etc., we would see with such a therapy, because the reversal, as you are correctly pointing out, is difficult. Currently, I don't know any antidote, so to say, for an siRNA therapy. I mean, it would be the gene overexpression, right, that you have to do, and this is this is now then gene therapy, so this is not doable. So you would have to deal with that hypotensive patient in a different standard way, so to say. So this is a really difficult question, but I think we have to see the clinical evidence and data and to see how many patients might develop, let's say, uncontrollable severe hypotension over time. So this is something, I think it's still an early time to say a final word on that.
Stefanie, any idea on that?
I fully agree with Thomas. I think if I would develop, if I would choose diseases, I wouldn't take hypertension as my primary, let's say, disease. I would bet a lot of money or would see that this is easy. I think there are other things which can be easier targetable.
Yeah. Carlo, do you have any comment?
Well, I was going to ask Stefanie, given her early interest in platelets, whether you see targetable targets in the area of thrombosis, atherothrombosis.
You know, these days I am now, it's 30 years since I was working with platelets. So I'm no longer the expert in platelets. I think, of course, platelets could be promising because they are in the bloodstream, so you can target them relatively easily compared to solid organs. So I don't think that this is undoable. I also know that in Mainz there are some efforts. I also see with Philipp Wild here, maybe he knows more, that Wolfram Ruff is developing or thinking about strategies to target platelets. But this is also not yet established clinically. It's not like targeting the liver, I would say.
Dr. Deveau, Ivan Deveau, do you want to ask your question directly? If you just show, we can unmute you. Ivan Deveau, if you look
it was not really a question, actually. It was more to try to enrich the discussion about the potential use of these RNA molecules as biomarkers, because I mean, not only us, but many people have done tons of different studies And we can see that the RNA molecules are detectable in the bloodstream, so that we can use as biomarkers, and they are dysregulated upon disease states. So we can very well imagine that we could risk stratify the patients by measuring the markers, the RNA markers in the blood, and treating these patients, the high-risk patients, with the RNA targeting molecule. And I believe that more or less what we call companion diagnostics, and that is an approach also that is developed in different companies, including in companies that are working on RNA therapeutics. So this is what I would call a theranostics. It's a combination of diagnostics and therapeutics, and I believe RNA molecules really have a powerful potential in this respect.
Yeah. This is only a comment. So we have Dr. Enrique Sanchez Delgado. You want to ask your question directly, or I can read it here? So if you show up, you made several good comments, and now a question. Dr. Enrique Delgado. No,
okay, if you listen to me.
Yeah, sure.
Do you hear me? Thank you. Yeah, my question is
almost
practical.
Yeah, you are not in the camera. Yeah.
Okay. My question is something very practical. We have still a lot to do with residual risk in cardiovascular lipids, etc., and that is lipoprotein A. We still don't have any good treatment for that, but there are good research and RNA-based substances that show promise to treat LPA. How do you think? How soon do you think can it be for clinical use approved, more or less? And by the way, I am very optimistic for RNA-based therapy. I did an analysis over 2 years ago, before the pandemic, and in the meantime,
we can't hear you anymore. But anyway, the question was clear. So maybe if Thomas want to take it, and Stefanie as well.
I can respond, and Thomas may add if he wants. So I certainly siRNAs against Lp(a) is very promising. It's shown to be effective, and clinical trials are running to see whether this has a clinical benefit. So it's not the question whether it's worked. It's worked as RNA. So do you see a reduction in the level? It's just a question whether you have consequences, because although Lp(a) is clearly associated with cardiovascular diseases, their causal contribution in humans has not been shown so far, because we had no chance to treat it, so to inhibit it. So I think this is really exciting. I don't know when the clinical trial ends. Maybe Thomas knows more about this, but I think soon. So at least it started already, I think 2 years ago. So looking forward to see the effect.
I share the same excitement. Also was eager to start, but my comments were exactly the same, Stefanie. So indeed, there are also several companies moving forward with siRNAs against lipoprotein A, and I also don't have a date in mind, but I also think the studies are over relatively soon. It's also a pretty large study, so we should see, hopefully, something or not, depending whether what the real importance of the LPA is, which is still debated. But it's super exciting. I'm really look forward to see what comes out.
Just to add, I think this is a fantastic example that you can use siRNAs to target something which hasn't been targetable before. And I think these are exactly the molecules I would and the pathomechanisms I would address with using siRNA therapeutics or other RNA therapeutics. I think this is if it works, it's a very cool example, exactly what we see as promising.
What do you think will be the most important challenges for the diffusion of the RNA-based therapies in the future? You think will be issues related to industrial production, or you said price cannot be a major issue, but maybe authorities or regulatory systems or ethical issues or people being reluctant to have when they hear about RNA they get sometimes they don't feel safe, like what happened during the COVID pandemic with the RNA vaccine. There was always confounded with the DNA, and at the level of population this could be another challenge. So what do you think would be one or more of these aspects the big challenge?
Yeah, maybe I can start. So I see more chances than risks in general, but it depends a little bit on the different fields. So if you speak about antisense RNAs, I mentioned earlier, so production, control of impurities, pricing because of cost of goods, production costs, etc. This will not be a big issue. I'm pretty convinced, and this will go easier and easier, and there are more production facilities also are developed. So first of all, the typical things are similar, to any other drug or compound you develop. So you have to check and control for side effects, and they can be, as mentioned by Stefanie, sequence specific. So maybe it's also another topic we have not addressed is to use really or to develop better preclinical models that can predict potential toxicities, etc. That's maybe also one thing that is very interesting currently, and is more and more developed, speaking about organoids and all these other new model systems. So I think there will be the biggest challenge, I think, and that was a question also earlier in the chat, is the, let's say, tissue-specific delivery. As mentioned, currently, liver, etc., is not a big issue. We have seen and we have own experiences with certain chemistries that a certain percentage, also by systemic administrations, end up in the cardiac. It also end up in other organ systems. So it's important to have the right dose that has efficacy in the cardiovascular tissue, but not has side effects in other tissues. So this is a balance we have to currently take, because so far there is no, let's say, clinically applicable, let's say, cardiomyocyte specific delivery tool. There are several things that are currently experimentally tested. If it's lipid nanoparticles or antibodies that are conjugated to RNA molecules. There are many different approaches currently, very exciting also. But this will be generation 2.0 also clinically. Currently, it's not ready for the clinics, but we will see this, I guess, in the next years, that this will also move forward, and that further will decline potential risks and side effects we probably may see in the development.
Massimo, to go back briefly to the issue of targeting Lp(a), just to inform the audience that about 2 weeks ago, Steve Nissen published a phase 1 trial in JAMA about this compound called lepodisiran, which is an extended duration, short interfering RNA targeting Lp(a). And he showed that you can get very profound, that is 95% suppression of Lp(a) levels in blood for as long as 1 year after a single shot of this compound. So there is proof of principle, but as Thomas said then, and as also Stefanie rightly said, we need to test the clinical efficacy of reducing Lp(a).
Maybe I would like to add to the question we had challenges. So almost I agree with Thomas, but I would like to maybe specify. I think the siRNA is targeting enzymes in the liver with a liver-specific delivery. We have a lot of evidence. There are 1,000 patients treated. So we have also data suggesting, at least to the time being, that this is quite safe. Of course, if we think about other strategies, if we need to target solid organs and increase the dose, or particularly if we think about using guide RNAs and CRISPR-Cas9 combinations for gene editing, then we have unknown effects which certainly need to be carefully watched out for. This may not be necessarily related to the RNA itself, like the guide RNA is also quite small and we have packaged them in nanoparticles, but maybe related to the gene editing, which can have potentially off-target effects somewhere in the body in other cells. So I think these are things where we just have first evidence that it works in principle. There are the sickle anemia treatments now approved. We have first patients for PCSK9 being treated with gene editing, CRISPR-Cas9 and guide RNAs. This is the RNA therapeutics. But of course, this is very early time, one has to be honest. These are just a few patients and yeah, we need to watch out carefully in the future what will be the safety profile. So I think we have to be very careful in RNA therapeutics is not RNA therapeutics. There are different types of RNAs, there are different delivery technologies and are different consequences. And we can also not mix the modified RNAs used for vaccination. It's not the same as an siRNA coupled to GalNAc delivered for cholesterol lowering. I think we have to be careful in not mixing these things and assess the safety profile one by one. And of course, we have to be careful in the future and watch all of this and need better models also. Thomas is also raising his hand. So
yeah, because I just think it's so important that you say that because, and I think Massimo, you mentioned that there seems to be a certain reservation in some patient groups or patients in terms when they hear something about RNA, and that obviously comes from the COVID mRNA approaches, although we shouldn't forget that we saved millions of patients their lives with very few side effects on the other hand only. I think this is very important to note, but this is a completely different type of let's say, chemistry and approach, completely different approach than with this antisense oligonucleotide approach. And the half-life basically is might be long, but it's still a transient intervention we are doing. We are doing transient interventions. It's not a gene correction or gene therapy trial where you have lifelong then a modulation of a certain target. It's still transient. And as I mentioned, this can be controlled by different chemistries. So for certain targets, you would like to have it nearly lifelong, yeah, LPA or hypercholesterolemia. So it's great if you have 1-2 injections only per year with a super long half-life. With some other things, and we talked about hypertension, I mean, it might be maybe valuable to have something that only has a half-life of a week or so, that you basically can play around a little bit also with doses. And these chemistries and a mode of actions, it depends also on the mode of administration, etc. I think this will develop and emerge so that we can also control a little bit better the half-life and the biological half-life of RNA therapeutics in the future. But it depends on the target.
Yeah, we have only 6 minutes, so I asked the audience if there is any burning question. I see there are other questions on the chat, but you know, if there is one of the participants who want to say a word as an important question.
Well, there was a question on NASH, etc., in the liver field. I mean, this is many groups are moving forward in the NASH field using siRNAs and other compounds. So there's very interesting targets, right? One is inflammation, one is fibrosis, and there are specific targets involved in those, and there are quite a few also clinical developments currently in this NASH field to move forward also as siRNA therapy. I just read one question.
Yeah, well, there's another one from Dr. Sobotka. He's again, this is related to the mRNA. So it's the mRNA vaccine myocarditis increase. You specify that this has little to do with the technology that we are using on the RNA-based therapeutics today. Is that correct?
Yeah, I think we had. Do you want to? Yeah. Okay. Good. Yeah. So I think we had discussed it just very shortly. There are different chemistries. I mean, if you talk about this, let's say, overexpression therapy using mRNA with the idea to overexpress something that usually are also very long RNA molecules that have a higher chance for side effects, in my personal opinion. In the cardiovascular space, there is one, at least I know one or two clinical trials where some biotech or pharma companies try to overexpress using modified RNA molecules, certain genes and then proteins later on. The problem really is the half-life and specifically also the delivery. Currently, there's a direct injection needed to get this into the cardiac muscle. And if you have a half-life of only a few days, then that kind of therapy might be the long term.
Stefanie,
this morning, Orna announced that it was cutting its staff, and this is one of the most innovative companies designing longer-lasting proteins that can be encoded by mRNA. I'm concerned, you know, we speak of the half-life of RNA itself. We should also speak of the biologic durability of the proteins that are encoded. I'm not sure what the strategies are for creating mRNAs that will encode for longer-lasting proteins, and who's working on them still now that a large player has sort of announced, by nature of its failure to secure funding, that it's, you know, involuting. So circular RNA proteins, great idea. What happened to the company?
okay, Stefanie.
Well, it's nice that you figure that out, but this I think only speculation. What happens to Orna? I think we don't know. I think the concept is really exciting, and that's not the only company that uses circular RNAs for protein production. But I think it's a bit a different topic now, I would say.
I would just like to point out that I think with the vaccination side effects, of course, it has been shown now that RNA-based vaccination strategies may have a bit more side effects than conventional strategies. But whether this is really due to the RNA itself, or whether this is really activating more profoundly the endogenous responses, or why we have some side effects, rarely, fortunately, it's still, to my best of my knowledge, not fully understood. So I think there is research needed to really understand this, and maybe then we can circumvent this. Maybe it's also biology, which we need to understand better, and we may also then find solutions for it. And I think there is a lot of ongoing work on this. With respect to the longer half-life of mRNA-based proteins, of course, this is interesting, actually. You can change the sequence of the RNA, and then you have a different tRNA used, and then you can get longer life. So you change a bit the sequence of the RNA, get a similar protein, but it's much more stable. These are things one can play around with. So it's interesting biology also, and we are just at the beginning. So I don't see the future so dark, but of course I'm also enthusiastic, maybe also biased.
So before turning to Carlo for the conclusive remarks, I would like to, first of all, saying that it's been a privilege and also very rewarding to coordinate this dialogue today. We thought it was going to be an interesting topic, and it is. We thought the people would have been more shy to ask questions on this topic, but it is not the case. And we had very good attendance, and people stayed through the end. So thank you very much from personally to Stefanie and Thomas, and to all the colleagues that have attended today. But, you know, Carlo, please, if you want to conclude the dialogue today.
Yeah, just to say that with Massimo, we were afraid that this would be an exotic topic that would intimidate the audience, but it turns out it stimulated the audience for a lively discussion. And this is due to the brilliant presentations and participation of Stefanie and Thomas. Thank you very much, Stefanie and Thomas, and thank you all for joining us in this very interesting dialogue. Also on behalf of Filippo Crea, who is the editor-in-chief of the European Heart Journal, we look forward to seeing you soon on the next dialogue, and you will receive information from Amelia Meyer, who is also behind the scene, a pretty important person we have to thank for bringing us all together on the web. So enjoy the evening and enjoy further reading on this topic. Bye-bye.
Thank you very much. Goodbye.
Thank you. Bye. Stay safe.